Hi friends, my name is Nandita. I am from Andhra Pradesh.
Children, now let us discuss about C4 pathway. Plants that are adapted to dry tropical regions have the C4 pathway mentioned earlier. Though these plants have the CO4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then, in what way are they different from C3 plants? This is a question that you may also reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate higher temperatures. They show a response to high light intensities. They lack of process called photorespiration and have a greater productivity of biomass. Let us understand this one by one. Study vertical sections of leaf on one of a C3 plant and other of a C4 plant. Do you notice the difference? Do both have the same type of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundle of C4 plants are called bundle sheath, and their leaves which have such anatomy are said to have Kranz anatomy, and Kranz means width and is a reflection of the arrangement of leaves. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick walls, impervious to gaseous exchange, and no intracellular spaces. You may like to cut a section of the leaf of C4 plants, maize or sorghum, to observe the Kranz anatomy and the distribution of mesophil cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical sections of the leaves. Observe under the microscope, look for the bundle sheath around the vascular bundles. The presence of the bundle sheath would help you to identify the C4 plants. Now study the pathway shown in the figure. Pathway that has been named the Hatch and Slack pathway is again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 acceptor is a 3-carbon molecule, phosphophenol pyruvate, and it is present in the mesophil cells. The enzyme responsible for this fixation is PEP carboxylase or PEP-case. It is important to register that the mesophil cells lack RUBS coenzyme. The C4 acid OAA is formed in the mesophil cells. It then forms other 4-carbon compounds like malic acid and aspartic acid in the mesophil cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release CO2 and 3-carbon molecules. The 3-carbon molecules is transported back to the mesophil where it is converted to PEP again, thus completing the cycle. The CO2 released in the bundle sheath cells enter the C3 or the Calvin pathway, and a pathway common to all the plants. The bundle sheath cells are rich in an enzyme ribulose biphosphate carboxylase oxygenase, but lack in PEP case. Thus, the basic pathway that result in the formation of sugars, the Calvin pathway, is common to the C3 and C4 plants. Did you notice that the Calvin pathway occurs in all the mesophil cells of the C3 plants? In the C4 plants, it does not take place in the mesophil cells but does so only in the bundle sheath cells. Thank you.